Okay, so so Lisa and I was essentially wanted to walk you through today in the next hour through how drugs are developed, and this is essentially thinking through or somewhat as presenting our perspectives on how medicines go from discovery all the way to having a molecular entity available at the pharmacy shelf and then making it, uh, accessible to patients from the early discovery phase. And again, this has more of the chemistry and pharmaceutical background, and I have experience working in the industry, so we'll be kind of giving you perspectives from those ends. And just to really walk you through these long, risky, and really pricey journey of developing a drug, the conventional track, essentially, you start from typically about 5,000 to 10,000 different compounds. This is again in early research discovery, where an interesting phenotype that you're trying to target, or a gene that tends to be, for example, upregulated in a given disease, and you want to actually look at how you can modulate that activity. So these 10,000 compounds are typically screened in mouse models or actually starting with cell lines. And really, simple questions are: what's the phenotype how can we modulate the phenotype that we know, and what is the molecular entity out of a given library that will have the most optimal activity? And again, this is somewhat iterative. You can synthesize multiple compounds, but you want to start from a large library to then narrow down, and then you go from 5 to 10,000 in the clinical phase. Once you've done all your couple of years of research in the preclinical phase, then you narrow down to about 50, and then once you go in clinical trials, you limit to 5, and ultimately only one of them make it to the market, and it becomes the available drug. And again, we'll go into details later on. But I just wanted to illustrate how long it takes. It's estimated that it takes about 12 years, and with an expense of over $1.5 million. Again, this can vary for just one single compound to become a drug that is used every day. And again, there's also the human cost, where there's a lot of people that have participated in these clinical trials from the early phase all the way to discovery. Ultimately, then the first stage is discovery, and I will let Liz discuss that more with a specific example. Liz, just let me know when you need to go to the next slide.
Awesome, will do. So in terms of discovery, so I wanted to start off with hopefully a more familiar cancer agent, Taxol. So the chemical name is paclitaxel. So we're going to go through a little bit of how it was discovered, the medicinal chemistry, and how we went from essentially a tree to the research and development bench, starting on the next slide. So I just wanted to give a brief introduction to what Taxol actually is. So it is an anti-mitotic agent. So in the most basic explanation, cancer is the abnormal growth of cells. And when you have an agent that stops mitosis or cell division, we're assuming that it will block cell growth. And so the indications for this, so I just listed this off from what the FDA label says. So first line, in combination with cisplatin, you'll see it used in advanced ovarian cancer, non-small cell lung cancer, and as an adjuvant or as an add-on, you'll also see this in breast cancer. And on the next page, so before I go into the specifics, I'll give a little bit of a history lesson, a background. So in the late 1950s, the National Cancer Institute, the NCI, they developed a department called the Cancer Chemotherapy National Service Center. So in the very beginning, all they did was look at man-made substances. And then in the 1960s, they partnered with the US Department of Agriculture. So now they started looking at natural sourced compounds. So they started going out and looking for plant-based and animal-based compounds that could be used for cancer agents. And so in 1962, they collected samples from a Pacific yew tree, Taxus brevifolia. So as you can imagine, the Pacific yew tree is mostly located in northwestern area of the United States. And if anyone is interested, Taxus is the genus for yew trees. So everything under the name of Taxus is a yew tree. Brevifolia, brevis in Latin is short, folium is leaf, so it literally just means the short-leaved yew tree. So in 1964, they realized the extracts from the bark of this yew tree were toxic to living cells, and they isolated the most cytotoxic one, which they called paclitaxel. So on the next page, they ran into a little bit of trouble, some ecological concerns that the extract is from a bark of a very slow-growing tree. So when they first were looking at it, you could only treat one patient with 6 100-year-old trees. And with a conservation status of nearly threatened, this wasn't a viable option. However, it looked like a really good drug. So what they basically did on the next page, so we do, as humans, we do what we do best. We try to synthesize it completely. However, the synthesis takes about 30 steps. That is not commercially viable. So we basically pivoted into a semi-synthesis. So we use precursors from the extract in order to make Taxol so it could be widely available. So on the next page, that's why on the FDA label for Taxol, you'll see it as Taxus bracata. So bracata just means fruit bearing. So if you look on the lower right-hand corner, it is a yew tree with lots of fruit. And this English yew, it's least concern in terms of conservation, it's widespread, it's easier to synthesize Taxol with, and it has a much more renewable source. So we're only using the pine needles and some like smaller twigs in order to extract the precursor. So why do I even bring this up? So in the next slide, here you'll see a list. So for one, if the total synthesis is 30 steps, it means that we are not currently at a very convenient step for making it completely man-made. So any master chemists out there, this could be a project. And in terms of precursors, so even in ancient times, lots of cultures have used yew trees for different types of medicinal purposes. And here is merely just a list of the yew trees that are here on this planet. So there's a lot of learning we can still do from nature. And now we're going to go a little bit into the more research and the discovery and research. I'll leave it to Libere.
All right. So now, when you have a drug of interest, and you want to know essentially, then how do you move it forward? And then this is really the field of discovery research, where there are multiple ways, both experimentally and computationally, you try to move your compounds of interest into the clinical phase. And one, the important things you can do early on is really to determine the binding affinity of your compound on the target of interests. And you also want to measure the biological activity that it has at different concentrations, because you want to determine the concentration that will be optimal for the patient. And ultimately, you also want to determine the mechanisms of action. So how does it actually modulate the target that you're interested in doing? And again, I here I'm illustrating 3 main known ways that the work is done. This is typically through what's known as phenotypic screening. What that means is you're doing a lot of experiments in model systems and looking at libraries of compounds. Or either libraries of compound or one compound, And even when you have one compound of interest, for example, in the case of Taxol, you still want to screen for other compounds that might be already existing, but they have, for example, the same chemical structure, and that you can hypothesize that potentially they may have the same exact activity. And so you want to compare actually how that would work. So you do this in both model systems, and you run screening assays on typically 96 well plates. Large companies usually have even, I think, 12 over 1600 wells. So very large plates where you can actually do highly multiplex experiments with many concentrations and many different molecules. And this is usually done with robotics that can actually monitor and very accurately disperse different concentrations. And here, you're able to determine, again, the binding affinity, the concentration, the optimal concentrations, and also whether there are other compounds that might be of interest. And you're looking for potential side effects, because if the compound is gonna bring side effects, you want to pick these up early on in the animal models. The other way that is done typically simultaneously is really target-based screening. So again, towards figuring out whether there will be side effects, you want to run the same compounds across different proteins. For example, you have genes that are in the same family, or they functionally do the same thing. You want to make sure that the binding of the molecule of interest is very specific. So you do this by essentially through target identification by in vitro and in vivo screening, to look at how well the compound modulate different complexes that involve your target of interest. And then computationally, in parallel, separately, you do structure-based design, where you can actually screen, again, libraries, or you can look at binding affinity, calculating binding affinity, or using the same molecule of interest across different proteins, or across different binding pockets, again, to establish the optimal optimal concentration by validating with either NMR or HPLC or other chromatography experiments that can actually help you, again, narrow down what the optimal compound is going to be. And I would just quickly share a few examples of projects that I've been involved with that were in the early discovery research. So this is a project I did in using structure-based drug discovery to identify inhibitors of an NS2, NS3 interaction complex in a dengue virus. So this was, if you look in the middle, the dengue is a virus of the Libere family, and it's a disease that is actually pretty bad because it actually breaks down your bones, and it's most common in the tropical areas. And the way it does this is it has a very high proliferation or high reverse transcription machinery that rely on these NS2, NS3 complex. And so what we were interested in was to figure out if we can actually use computational structure-based screening to identify some compounds that can actually inhibit the binding of these two proteins by essentially fitting into the pockets where they typically fit. And so I did some computational modeling to actually identify that there were these two specific pockets in the structure that were driving that interaction. And it was a tryptophan and a valine pockets. And so what I did, I actually screened about 7,000 different molecules. And this was after I did computationally to identify the top compounds that had higher activity or higher binding affinity for each pocket here. So on the left, then we did in vitro screening experiment to look at what's the protease activity for each of these pockets. And as measured by the IC50, when you look at every every one of the top compounds. So on the y-axis, essentially that's the IC50 percentage, and IC50 is the concentration at which the compound is able to inhibit the activity by 50%. So when you have lower, that means the compound is highly active. So it's limiting the protease function. And when it's higher, it just means it's lower. And so, and then you can plot that, and then this essentially help you establish which compound will be optimal. And some of these were moved into cell lines, and ultimately in mouse models. Another project that I worked on was really identifying, again, binding compounds that would modulate SGLTs. These are sodium glucose co-transporters, transmembrane proteins that are involved in transporting your sugars in the kidney. Obviously, these are of interest to disease such as diabetes, where you want to ask the question whether or not, if you were to modulate this, you can reduce the amount of intake or sugar intake that is taken in by the SGLTs. So one part that we had to do was to actually model the structure of the protein, because what was available at the time was a bacterial version, a 3D structure of the bacterial homologue of the SGLT. So what I did, based on this and looking at the sequence homology, you can actually computationally model the 3D structure of what the human version will look like. And then after that, again, run docking experiments to look at what molecules are out there, are available, that can bind in the same exact pocket where the sugar binds, and the idea that they will be competitively binding at accessing that pocket. And if your molecule is more efficient at that, then it will be a good inhibitor. And so this is essentially showing how these three ligands were very nicely binding into the exact pocket that the sugar binds, and these will be ideal candidates if you're looking for compounds to be inhibitors for SGLT. The other part is actually for this is that if these compounds are good, have really good affinity, they can also be used to crystallize the human version of the protein, because it allows you to fixate the molecule in a three-dimensional conformation, and you can crystallize it. And so you have actually a human crystal structure of the SGLT. Similar to the previous two, this was another different project that I worked on. And the interest here was to identify, again, inhibitors of these disaccharide derivatives. These are, so the lab had identified these disaccharide derivatives. These are just different molecules that that are all derivatives of sucrose. And what they had done experimentally, they had shown that they actually reduce biofilm formation in bacteria, and they also reduce cellular motility. And what that means is that, so for example, in cystic fibrosis, in the lungs, you have bacteria that grow and form a biofilm, and that's how they proliferate. So the idea was, if these compounds can actually inhibit that the biofilm formation, then we want to know if they can be used as drug targets. So computation here was looking at how each of these different compounds are binding to the flagellin protein, which helps those bacteria actually interact. And the idea was, if we can find the most optimal inhibitor, then that would actually be useful for as a potential therapeutic molecule for. And then Liz jump into the preclinical research.
So for preclinical research, I did more of the definitions. I didn't know how much everyone already knew of it, but essentially you have two spheres of where you would do research, in vivo versus in vitro. Vivo in Latin means to live, therefore it's inside a living organism, while in vitro is the exact opposite. It's all the test tubes and cell cultures that Libere has talked about. So a couple types of preclinical research. Toxicology is probably the most self-explanatory. You want to know what the lethal dose is, what at what dose will you have adverse side effects. The next two, I'm going to go a little bit more in detail on the next slide. So the difference between pharmacokinetics versus pharmacodynamics. I just kept this really simple, but kinetics simply is the movement of the drug within your body. So the question we're asking is, so what does your body do to the drug? You can kind of think of it as food, right? So you need to break down, you need to digest and absorb food. While dynamics is the exact opposite question. So what effect is the drug have on your body? So the effect of the drug and the mechanism of action. So in the next slide, I talk a little bit more about what pharmacokinetics looks at. So ADME, well, you also see this as LADME. So L would just be liberation, so how the drug gets released from the dosage form. And then when you go into A is absorption, so how does the drug go into your blood circulation? Distribution, how does it go from your blood vessels into your target tissues? Metabolism is how it could be broken down, how it can be chemically changed for easier elimination or not. And then finally, excretion. So how do these drugs leave your body? And the chart that you see on your right is kind of the basic chart of how we look at drug exposure in someone's body. So everything in the green, absorption. So all of that is the drug going into your body, reaches Cmax, highest concentration, and then everything after that is elimination. And why there's a half-life is because we want to know how long the drug actually stays within your body. And this will affect how much drug is given as well as how frequently you're dosed. And so in the next slide, I talk a little bit about pharmacodynamics. I know that I probably should have used a cancer agent, but I wanted to talk about desired activity versus undesired activity. So desired activity is your effect, right? While undesired activities are side effects. So Revascio, originally, the desired effect was to decrease pulmonary blood pressure. So it was made to be a cardiovascular drug. So what it does is it decreases the activity of your phosphodiesterase 5, which causes the relaxation and widening of blood vessels within your lungs, and that helps bring down the blood pressure. Some of the side effects included headaches and heartburn. And so these are side effects, right? These are not what this drug intended to do. However, on the next slide, there is one more side effect that it had that they found, which is it increased blood flow in the penis. So at this point, some of you might already know what I'm trying to get at. So this Revascio is also known as sildenafil. And on the next slide, it's also Viagra. So essentially what your desired effect versus undesired effects also depends on what you're targeting. So remember, be a little bit flexible in what you're looking at, because you never know which compound will also create a new drug.
And so, and I wanted to go quickly just through the different phases as well. Again, phase 1, you want to determine the safety and the dosing. Phase 1 is usually done in about 30 patients, and these are typically known as small proof of concept. They are just fewer patients, it's cheaper, it's a lot faster to do, and it's also, and at that point you also have just a small set of molecules you want to do, and it allows you to generate really initial solid data to be able to submit to provide to the FDA so that you can move into the next phase. And phase 2 is essentially most of the time is typically just is separated into 2 phases. In A, you're expanding what you did in phase 1 into a larger cohort of patients. Again, to demonstrate that the efficacy that you showed in the phase 1 is also still solid when you had a larger cohort of patients. And again, this is narrowing down your number of compounds you want to pursue. And ultimately in 2B, then that's where you do dose finding to find optimal dose in humans, and also in a much larger cohort of patients. And Liz?
So phase 3 clinical trials is basically a buildup of phase 1 and phase 2. So now you're looking at a compound and you want to determine the efficacy as well as monitor for adverse reactions in a much larger patient population. In general, so if you're doing, for example, like a blood pressure medication, the patients will be 300 plus, 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 plus. So you could have multi-center, even potentially multinational, especially with how pharmaceutical companies are run nowadays. And so you're trying to evaluate an overall benefit versus risk. So unless this medication is for a condition that has no treatment, or you're targeting a very specific population, you usually want to compare the safety and efficacy of your new drug to a current standard treatment. The reason why we want to do this is, honestly, there is no real value in having a drug that does the exact same thing with no added benefit to what is currently on the market. Of course, there are a lot of drugs that are like that. However, one of the things we do want to evaluate is what further value can we bring to the patients. And then, so the FDA review, so the Food and Drug Administration, they are consumer-based. So they want to make sure that anything that they give approval for will benefit the patient more than harm it. So there are two types of applications. So there's an IND, an investigational new drug, while an NDA is a new drug application. So on the next slide, I talk a little bit about INDs. So INDs, there are two categories. So one is commercial and one is research. Those are a little bit self-explanatory, so I'll go into the three types of INDs. So INDs are generally, you want to do this around like phase 1, maybe before phase 1 or after phase 1. So anytime before you start looking at like human population, essentially. So the three types of INDs, you have an investigator IND, in which you have a physician who is in charge conducting this experiment, and they're going to be the main person who is going to be administering and or dispensing this medication. And generally, these can be more research-based in the sense that you are looking at the efficacy and safety of an unapproved FDA drug. While emergency use IND is exactly how it sounds like, it means that there is an emerging situation in which a full-blown IND process would take too long. So they're trying to expedite the process. And then finally, treatment IND is essentially that in clinical testings, you are seeing promise in the efficacy. And so while it has to be kind of a serious and life-threatening condition. So while you are finishing up your clinical trials, the FDA will be reviewing it at the same time. So the next part is where I talk about what an IND needs to have. So animal pharmacology, toxicology studies, these are just to prove that once you start initiating this treatment, this agent within humans, that you are saying it's reasonably safe to start this. Manufacturing information, CMC, chemistry, manufacturing controls. This is usually to reassure that you are able to produce this medication at a consistent strength, quality, and purity. Clinical protocol and investigator information is pretty straightforward. How are you going to be testing this drug? What populations? How are you going to enroll? Your inclusion, exclusion criteria? What is this experiment powered towards? And what are your endpoints? And investigator information is just to prove that the person who's running the experiment is qualified to do so. So in the next slide, I talk about the new drug application. I think the FDA puts it best. So I just took it directly from the FDA. Essentially is when a drug sponsor, it could be a manufacturer, any kind of money that's put behind it, that they formally propose to the FDA that this drug should be approved as a new pharmaceutical for sale and marketing in the US. So now this becomes commercial. So your IND data generally becomes a part of your NDA. So on the next slide, so an NDA basically asks some of the same questions. So is this drug safe and effective for its intended use? Does the benefits outweigh the risk? We see this from phase 3, but the other two, so is the drug labeling and the package insert appropriate? So now you have to paint a picture. You have to put together the story of how this drug works, why it works, and why it should be essentially put onto the market. And then finally, of course, they want to make sure that your manufacturer controls are in place so you don't get inconsistent batches, that it's commercially viable for this drug to be on the market. And then on the next slide, I basically just did a comparison between an IND versus an NDA. So an IND, you're still answering questions about drug safety and efficacy. So does this drug work, and what are its benefits? While a new drug application, you're looking at the value of this drug on the market. So the FDA is going to start looking at, so if we market this, what value does it bring? And so one is you're still building the story, and the other, your story is ready and now you're packaging it. And for the final, there is a final phase. I'll leave it up to Libere.
Yes, so as you can imagine, although the communications with the FDA is really a lot of document, but ultimately this is critical again because FDA is a patient-centric and market-centric entity. And the final aspect of a drug, once it's been submitted, gone through all the phases and approved by the FDA, is the monitoring post-approval. And there's an entire division at the FDA that is in charge of surveying all the drugs that are on the market. And really, this is of interest both for the FDA and for the companies themselves. So, and for the FDA, the importance is to monitor different adverse events. This is for the interest of patient safety, and also to continuously monitor that the proposed efficacy and the approved efficacy for the drug is sustained as you expand the outside of the clinical trial context. And finally, the reporting problems can be submitted to the MedWatch, but most of the time, most of companies do have a team that keeps track of all the adverse events that are being reported. And most important, this is really good for the companies and for the patients ultimately, because this is how you find new indications. And drugs can be repurposed. For example, in the case of Taxol, which was probably originally submitted for a given cancer type, you see that now it's used for both breast and lung. And that's because at some point, as part of, again, this monitoring post-approval, was found that it could have an activity in a different set of patients. And an even more better story, it would be the Revasio story that was first approved as a cardiac disease drug, but then men started going into the hospital saying, "Hey doc, I have this thing, this weird side effect that I'm observing. I don't know why." And then you see, as part of the post-approval monitoring, they saw that all the men were reporting the same thing. And so I believe it was Pfizer. Pfizer was like, this is probably something we should look into. And so they went back and then did, by monitoring what the biological activity they are observing as part of the adverse events reporting, they realized that there was another indication. So they pursued the same exact drug as an erectile dysfunction compound. And now it's a multi-million-dollar drug, again, as a consequence of this post-market approval process. So, and I think that's the last piece of what we had, and it's perfect. We have 16 minutes for Q&A. Thanks. Feel free to shoot questions for Liz and I.
Hi, Libere. I just had less of a question about more of the science stuff, but like from your experience in being in industry and working in it, what do you probably say is like the biggest shift into academia, and like either in how you work or just like what the work you do is?
Yeah, good question. So so in industry, generally, I would say things are faster. Things are a lot stricter in the sense that it's money, and you're trying to move things as fast as possible, and projects are defined by what's marketable. And what that means, although in the research discovery, in discovery research, there's plenty of discovery projects you can do. But they all, typically revolves around the biology of the disease, and then how you can modulate that biology, and then how do you find therapeutic opportunities? While in academia things are a lot slower. You can hang out as long as you want, and it's just all chill. Your project, you can expand your project for almost as long as you want. So to me, that aspect has always been crazy. I'm like, why is no one moving this project fast enough? And with that, in industry, projects really die quick, right? If a molecule does not have really, really impressive activity, it will be killed, because you don't want to spend $1 million on that project. And so the turnover on what you can working on is quick. You can work on 5 projects in 1 year. But in academia, you're essentially moving, taking your time, moving slowly on the same project for 3 years. You can be working on the same thing. That's one aspect. The other aspect, personally, I was working in a team of about 30, 35 people, and they were all. This is one of the reasons I actually came to get my PhD. I was the only one who didn't have either a doctorate or a master's degree. So I went to get a PhD because if you want to actually contribute to generating new projects, you want to have the deep expertise, the deep knowledge. So the people are typically really, really smart in industry, in the sense that they're super adaptive, and they're a lot more creative. And I think that has to go with the culture that you have to be innovative and fast and ready to go. While in academia, you can spend your entire career studying one gene because you want a Nobel Prize. I think that's kind of one way to show the difference.
Thank you.
Mm-hmm.
Other questions, you guys?
If no one else has an, oh, Nathan, please go.
Oh, okay, yeah. Libere, you're talking about discovery research in terms of phenotypic screening, target-based screening, and structure-based design. So does that mean all these 3 happen in parallel to get to narrow down the best compound?
Yes. So typically they happen parallel. The structure-based screening is usually done by computational teams.
Okay.
So usually you have an entire computational team that's doing the structure-based screening. And in parallel, you have a biology team that's doing the phenotypic screening. And again, this is if you're working for a large corporation, obviously.
Yeah.
If it's a small startup, then it's usually just 5 people that are just working 80 hours to make it work. But ideally, in the large corporations, we usually have 2 separate teams, a biology team and then a computational team, that are looking at the same compounds at the same time, and then talking to each other, almost meeting every single day. Because they have to be fast, quick turnover. Because if you want to go from 250 compounds to 1 or to 2, then you have to do a really fast elimination process.
I see.
So yeah, so usually it's 2 teams that are going at it separately. 1 more computational, and then the phenotypic screening is done by the biology.
And what's the difference in the outcome between the compounds identified by these? Are they vastly different?
No, so typically they're the same. So the structure-based design is looking at the predicted affinity, right?
Mm-hmm.
Because you're just doing computational modeling, and you predict affinity, and you want to rank out of, say, 200 compounds, which are the top 10. And then your top 10, and you bring the affinity, and then you typically want to, you also want to show in 3D how each of the compounds is
Mm.
actually interacting with the amino acids on the protein. And
Okay.
then you bring that to the biology team to say, hey, this is what our models are showing us, right? It seems like this compound is better because it's interacting with the tryptophan and the valine, and this is what the literature says that it seems to be how the protein functions. And then the phenotypic screening group goes and then maybe knocks down that protein and then treat with, you have two experiments where one you knock down the protein, other one you don't, and then you treat both with the compound, with the different compounds that the computational teams suggested, and you can see that you validate what they suggested. Right?
I see.
And then once you validate, and then again, you're looking at all 250, also in the screening experiment, and then you kind of cross-validate each other.
Oh, okay.
Yeah, and then you go to the PharmD team, the clinical team, and then Liz is like, that's cool, but I want to see the kinetics. And then you send that out to a chemistry team that do the pharmacokinetics and the pharmacodynamics.
I see.
So it's usually like many teams that are kind of going at each other, arguing scientific arguments
Yeah.
in favor of one drug over the other, and then they go on and on and on, and then look into the literature and then confirming.
Mhm.
Yeah.
I see. Thank you.
Mm-hmm. Sachin?
Yeah, so I was just gonna ask, so with the different like phase trials, I'm figuring that like the application or just the testing of it would probably be a bit like a bit overlapped, considering that yet they do have different focuses. Do you ever like in like a phase 1 trial end up addressing things that would be applicable in a phase 2 or phase 3, or is it like simply what you're measuring just safety and efficacy, or just safety and like not trying to touch like significant efficacy or anything like that?
That's a Liz question.
So currently I'm working on a trial, and essentially you can look at things that you can look at efficacy, you can look at safety for further trials. However, that is not what the FDA is going to accept. So for example, for phase 2, we really only want to look at the optimal dosing. We really want to look at just the safety side of it. So you can document, so in your endpoints, instead of to determine the endpoint, you have to just say to document the X, Y, and Z, and it can't be your primary. So yes, you can look at that. However, it probably won't be powered the same because you won't have enough patients for it. And the FDA definitely, the first thing they're looking for is whatever is required in each trial. So yeah, you can definitely look at it. It just won't be something that the FDA will accept as like a phase 3 decision, like a phase 3 determination. I don't know if that answers your question.
Oh no, it does. It does. I was just wondering, like, if you could potentially do like successive phases almost at the same time. But yeah, I mean, it makes sense if not.
Yeah, just to add, so different companies actually are interested in doing that, right? Because that saves you money.
Yeah.
If you can do all this early on, then you save money. For example, Novartis, they've developed this new model of drug development where they do proof of concept in animal models before going into humans. They use, for example, primates and dogs. And you can do all these studies in animal models before you even go into the humans. And you can look at everything that you would look at in humans, but look at it early on. Mm-hmm. Because it's good as data you present to the FDA, although it may not be what the FDA is saying, as Liz said, but it's also good for you to know ahead of time, right? Before you invest, $5 million. If you can get the data you need before, then it's also good for you as an assurance.
Okay. So does that would all fall under like that preclinical testing?
The preclinical, yes, the preclinical phase.
Yeah, just to add on, the FDA only starts caring when you go into humans. You start doing human trials. So beforehand, pretty much, you can look at anything and everything you want.
Awesome. We have time for about 1 or 2 more questions. We have 5 minutes. If you guys have any more questions for Libere and Liz. No?
I
Okay.
mean, I have another one, but I'm like hogging all of the time, so I will
No,
definitely
go for
open
it.
it up. Does anyone else have anything? Okay, well, my last question was that with respect to, because I've heard to in a certain capacity that physicians are often consulted by companies like actual doctors. In what capacity have like you seen that, and how does that really work? Because I've only heard it just like dropped as a name, like, oh, I consult or I talk for this company. But how does that really like manifest?
I'll answer first and I'll let this answer. Novartis has an entire translational research team, and they're all MDs. So on every trial, on every ongoing trial, there is an MD person on that team, because you need the doctor's perspective. And that's not even including the outside consultation, but they have an internal MDs that are hired specifically to bring in the physician perspective, and they all contribute to say, in determining to say, this group of population will benefit, this group of population will not benefit. Here's what I already know about this class of drugs or this class of molecules. And things like that.
That was a perfect answer. Pharmacists and doctors, we both can be either consultants or part of the internal team. Essentially, if you're a part of the internal team, as Libere said, that you are actually actively working on it. If you're consulted, I had quite a few pharmacist mentors, who basically they would get these cases and they would evaluate them and see on a pharmacological perspective, does this make sense? So essentially consultants work as a consulting.